Welcome to this Headwaters Science Center 3:30 show. I'm glad you guys are all here with us because I just stumbled upon a super interesting crime scene. Looks like we have multiple vehicles and a strawberry involved. Oh my goodness. Cars are backed up, piled up, and it looks like all because of this strawberry that got crushed. Oh my goodness, how are we gonna be able to identify this strawberry? Let's think back. When I remember watching crime TV shows, I think they're always trying to identify people on DNA. Does a strawberry have DNA? Well, of course it must have DNA because it has cells. If something to be considered a cell, you need to check off a few boxes. And one of those boxes is having DNA. DNA or deoxyribonucleic acid is the genetic information of our cells. It basically tells our cells what function to perform or what things to make. So let's see if we can get some DNA out of this strawberry. All right, so what you're going to need today, if you want to do this experiment at home, all you're going to need is 2 or 3 strawberries. Prune the leafy green parts off. You're going to need a Ziploc bag. You're going to need some soap, dish soap, a small beaker, another small beaker, and teaspoons and a measuring cup, and a stick. And I think the last thing is going to be isopropyl alcohol. So first step is the DNA is located in the nucleus of a eukaryotic cell. Strawberries are eukaryotic cells because they're multicellular and they have membrane-bound organelles. So the best way we can start this process is just by physically mashing them up. We got to try to create separation in between those cells. So mash them up in your bag. Do it for 1.5 minutes, 2 minutes. Make sure it's an even consistency. There's no lumps. And you want to mash it up as much as you can. Separate those cells. And you said you put 2 strawberries in there? 2 strawberries. You can do 1, you can do 3. I just picked 2 because it was a good even number. Alright. I'm going to mash it up a little bit later, but I'm going to move on to creating another solution that helps break down the cells further. So last week we used bubbles to represent cell membranes. And if you remember why we used that bubbles to represent cell membranes, it's because the soap has a hydrophilic head and a hydrophobic tail, which is just like a cell membrane. Another good thing to point out that dish soap has a hydrophilic head and a hydrophobic tail is that it helps dissolve hydrophobic substances like grease or other fats and lipids. Because of that, because it has that hydrophobic tail and hydrophilic head, it's able to bond to the other hydrophobic areas on the greasy pan or whatever. And that's kind of basically the same thing we're going to be doing, but we're going to be mixing it with cell membranes. Every one of these cells in this bag is surrounded by a cell membrane. We're going to use Dawn dish soap to help break open that cell membrane and expose that cytoplasm. All right, so the recipe that we're going to be using today, we're going to need a teaspoon of salt. Salt?
Is that a teaspoon?
Tablespoon. There we go. Tablespoon. Tablespoon of salt. And 2 tablespoons of water. Or soap. these salts help help break down the proteins that could be bonded to the DNA and help isolate the DNA once the soap has separated the cell membrane. Then we're going to need half a cup of water. I'm going to do cold water just because it works better if the experiment stays cold.
And then a lot of the experiments we do around here, we use reverse osmosis water, but this is fine to just use tap water for?
Yep, just any tap water that you had at your house. Alright, half a cup going in there. And then you're going to want to stir that up. You want to stir it up gently. You don't want to create bubbles as best you can. It may be difficult to get all the salt dissolved in it, which is okay. Just do the best you can. Alright, so we'll let that stir a little bit more.
So you want to get the salt dissolved in your
Yep, you want to try to get it
solution
dissolved a little bit as best you can. And you're not going to get everything. Once you've mixed it up, stirred it up for a while, go ahead, find your mashed strawberries and put your solution, your bubble and salt solution, into your bag. Alright, and you're done now with this jar, so you can put it off to the side. And you zip up your bag again and mixing. You're going to mix it up again. Smash it gently. You're going to try not to get bubbles. Continue to mash it up. Mix the salt and soaps into these strawberry cells. You go ahead and do that for about a minute.
So you said
Or so.
Soap is helping to break up the cell membranes of the strawberries.
Yep.
And what is the salt doing?
The salt helps free up the DNA and break down or separate it from the proteins that could bind to the DNA. So all right. And then our last few steps here are going to be, you're going to need a beaker and a coffee filter. Coffee filter helps, we're going to strain all the large chunks out, just looking for the liquid part. And this part kind of gets tricky if you're all by yourself. So science is always better when you're doing it with a partner, with a lab partner or a family friend, a relative, anyone. Scientists today that are studying anything in the whole entire world are never working alone. They're always working with partners, communicating, throwing ideas back and forth. So a big part of being a scientist is being able to work effectively with other people. All right. So we're gonna strain out all the liquid stuff. The DNA is gonna be small enough to pass through this semi-permeable membrane. So I'm gonna try to get it in here without spilling all over. Oh, we got it. Whoa!
Maybe not.
Try again.
I got your coffee filters.
Yep. Try again. Okay. And you do again. Oh. A little bit.
It might help to have a bigger beaker here too.
Oh yeah, it did fall out. Alright. I'll rinse out this. Alright. Try again. Third time's a charm. Alright, one more coffee filter. I'll use this medium one. Alright, I'll just try a little bit at a time there.
Kind of smells good.
Yeah, it smells like soap and strawberries. Alright, kind of got this filter full, so let this drain. You're, again, trying to restrict all the thick fibers, other structures of the strawberry cell, and just letting the small particles, the DNA, the sugars, anything that's able to dissolve in that liquid, pass through. And so you can gently squeeze it. Try not, don't squeeze too hard because you might rip your filter. But put a little bit more in.
A little messy.
Yeah. Alright, I think this is going to be good enough for a large enough amount for us to get some results. Alright, then the last factor is going to be the isopropyl alcohol. So you're going to want to keep this really cold as you can be. And you're going to want about almost a half a cup.
So you're using the 92%, it looks like,
Yep.
there, rather than the 70%.
From what I've seen, 91% isopropyl alcohol works better than the 70%. It seems like they both get results, but you get more results with the 91. So then you're going to gently pour down the side of the glass the chilled isopropyl alcohol. Oops. Oh.
He just had his stashed in the freezer here at the...
And you don't have to worry about it freezing because 91% alcohol would need to be quite cold to be able to freeze. Alright, so within seconds you're able to see a separation between the blue, kind of purplish blue, white bottom and this little thin layer on top of it. And let's see, now you can use a stick or use anything really to go ahead and swirl around, collect some of that goo,
Getting the stuff off the bottom.
getting the thin layer on the top, just above the white.
Getting that between layer.
Alright, right there. This goo that comes out should be your, should be,
Put it lower, put it in front of your shirt.
okay, should be the DNA of the strawberry. Strawberries are really good at this because they have a lot of DNA compared to other organisms. We in our cells, we're diploids. And strawberry cells actually have 8 sets of DNA, where we only have 2 sets of DNA in our cells. So that just means that they pack in more DNA than other cells. So that's why it makes it such a good way to experiment with DNA. Let's see, can I get any more? So it's kind of this booger-looking substance. So DNA is made up of nucleotides. Nucleotides form together to make nucleic acids. And when you bond together enough nucleic acids, you get DNA. Depending on how that those nucleic acids are actually sequenced, it will alter the information that DNA reads. So that's basically where the proteins in your cells read the DNA, saying, "Oh, these nucleic acids are in this sequence. We're gonna perform this function."
So that's why when the nucleic acids are like the letters you see associated with DNA, right? Where it's like your T and your C's and your G's that are.
Yep. Awesome. I have one that's here earlier. It sat out. I don't know. It seems like once you let it warm up, the DNA actually denatures. You're not able to collect it. So having it cold definitely helps. All right. Well, thanks for tuning in to help me solve this strawberry crime scene. I'm going to take this DNA to the lab to really investigate this strawberry further. If you want to come on down and experiment with DNA firsthand, come on down to the Headwaters Science Center any day of the week. Monday through Saturday, 9:30 to 5. And then swing by after lunch on Sunday from 1 o'clock to 5 p.m. All right. Well,